Sorry about that. Thanks for the invitation. And today I'm here representing Stomics. I'll walk you guys through what exactly is Stereo-seq, and it's offering a spatially resolved cellular omics solution to your research. So for expert scientists like you, I'm sure there are a lot of advanced tools and technologies that are available in your lab already that are providing solutions to address your questions and research, but there are still many questions that are left to be answered, like the spatial context of the tumor microenvironment and how does drug response to the tumor and immune cell crosstalk. So what are the solutions to that? And this leads to the question of why do we choose spatially resolved transcriptomic sequencing solutions? And I think for us to be able to answer that, I think everyone knows that location matters, and locations matters in a way that can answer your questions about providing insights and allow you to dig deeper into the study of tissue biology. And so compared with traditional single cell sequencing and bulk sequencing analysis, spatially resolved transcriptomic sequencing is offering a new way for you to bring, in a never way before, that you will be able to see gene expression profile in a spatial context at a very high resolution. And I think for Stereo-seq, it's offering a way for you to discover. This is made for scientists like you to make breakthroughs and to dig deeper into the discovery aspect. So to provide cell location information within the tissue section, I think Stereo-seq can offer the importance of identifying the cell location, as well as telling a fuller story about the state of the tissue microenvironment, which plays an important role in disease progression, tissue microenvironment, and also in drug discoveries. And in this case, we're using Stereo-seq technology to study the kinetics and directionality of transcriptional variation in a time course within a mouse embryo to study the mouse organogenesis. So for spatial omics sequencing technology, there's still so many challenges that we're facing. And there are two major challenges that we face today for researchers like you, like the low resolution and the small capturing area still remains a hurdle that we aim to strive to overcome. And for Stereo-seq, we are designed to tackle these two problems, and we are we're one of the highest resolutions in the market, also providing a centimeter field of view that's we're indicating that Stereo-seq technologies here definitely outperform some of the existing technologies in the market. So Stereo-seq is the one and only spatial omics sequencing technology in the market that can achieve both centimeter level of field and also a nanoscale resolution. And this is independently developed by Stomics. We are based on DMB-seq technology, providing a panoramic field of view for your research. So what exactly is Stereo-seq? Stereo-seq stands for Spatial Enhanced Resolution Sequencing, or omics sequencing. So it's based on DMB-seq technology and operates on a unique principle to unravel the spatial organization of gene expression within biological samples. So Stereo-seq chip is based on DMB-seq technology, where we load on DNA nanoballs onto the platforms, and these are really, really tiny DNA nanoballs at a size of 220 nanometers with the center to center distance of 500 nanometers. And these spots on the chips are composed of CID, which is a coordinate ID that can identify the spatial location of your mRNA transcript, and also with a unique MID that's tagged to the molecular portion of the transcript you're interested in. So what Stereo-seq set apart in spatial transcriptomic technology is that it offers subcellular resolution, which is one of the highest in the market. And to demonstrate the robustness of Stereo-seq, here we prove its ability to dissect tissue sections with cellular resolution. So here we profiled a mouse coronal section with Stereo-seq, and when zoomed into specific regions, you can actually see aggregates of mRNA transcripts with the DNA nanoballs that are captured. And additionally, we're combating the low capturing area problem in the field by offering a large field of view with the efficient capturing of by the whole transcriptome of your tissue. And here we're demonstrating the flexibility of it and also the efficiency of obtaining the information of the entire tissue section without having to score the tissue blocks. And here is one of the examples we're showing a macaque cortex, a macaque coronal section that's displayed on a 3 centimeter by 5 centimeter Stereo-seq chip. And I'd like to walk you through some of the applications, the publications we've done. And so we have some of the displays that people, you can hold it in your hand and take a look. So in an early publication by Cheng et al. In the journal Cell in 2022, Stereo-seq overcame the limitations of existing methodologies, striking a balance between capture efficiency and resolution and also field of view. So the study sheds light on the molecular basis of spatial cell heterogeneity and cell fate identification in developing tissues. So we were able to build a cellular resolution Stereo-seq atlas of mouse organogenesis and to review the spatial diversification of cell types at whole embryo scale. And additionally, with Stereo-seq, we were able to construct world's first spatial temporal cellular atlas of the axolotl brain. So this is a brain of an animal called salamander, and they are known for having large genome size of 32 gigabytes. So it's usually really hard to process these kind of genome scale data. And with Stereo-seq, we offered high resolution and large field of view at the same time. Additionally, we're providing tools that be able to process these high throughput data. And on our spatial transcriptomic data highlighted the cellular and molecular features of the axolotl brain regeneration process. And we were able to identify some of the specific neuron stem cells and also the activation of functional regulations of them within the regeneration process, and specifically an injury-induced regeneration process. And this can let us to discover some of certain glial cells within the tissue section that may be improving the regeneration capabilities of mammalian brains in the future. So besides on tissue regeneration and organogenesis, Stereo-seq also has impactful applications beyond developmental biology, notably including cancer research. In a recent publication by Wood et al. From MGI research, they were able to dissect the cancer ecosystem through characterization of the spatial transcriptomic heterogeneity around the solid tumor, and specifically at the tumor border. So they were able to gain a comprehensive understanding of the complex tumor environment and the cell-to-cell interactions at these border lines. And this is important because for hepatocytes, it's usually a type of cells that we wanted to study in deeper, but usually could not be achieved with single cell sequencing because hepatocytes tend to not work well with single cell sequencing, like the dissociation. So by capturing the molecular characterization and dynamics of this solid tumor environments, we're able to provide insight into the tumor ecosystem and further to propose what could be the invasion path of solid tumor in this context. And there's another great example demonstrating the ability of Stereo-seq. So recently in a comprehensive paper that we published, we profiled 161 coronal sections of a macaque brain cortex at a size of 3 centimeter by 5 centimeter. And in total, we segmented over 100 million cells that are collected from all the sections, and we're able to produce a comprehensive 3D single cell atlas for the cortex of the macaque brain. So what did we learn from this paper, and how did Stereo-seq help along the way? So Stereo-seq reviews the global distribution of 264 cell types and their marker genes. Scientists were also able to discover the regional density and composition of cell types are actually coupled with cortical hierarchy, meaning that many of the cells regulated in a manner dependent on the hierarchy in the cortex. And lastly, they were also able to do on cross species. They evaluated the cortex the cortex of the human, the mouse, and also the monkey. The analysis reviewed the primate specific markers and cell types enriched in a specific layer, and their marker genes expressed in a region-dependent manner as well. So if you're interested in getting to know more about the data, they actually have an open access data set available online for you to browse around to learn more. So how can Stereo-seq impact your research, and what kind of applications do we offer? So currently, Stereo-seq, we're providing kit solutions all around the world. And this is our first time presenting Stereo-seq in Korea in public. And so I would like to talk more about what exactly we offer in our pipeline and the workflow. So for the kit and reagent solution, we are offering easier user adaptability and also improve user experience with our newly designed kit. So our workflow has also been optimized with specific biochemistry design for more efficient cDNA collection along the way. So what does the Stereo-seq workflow look like? So it's really simple. So you pretty much attach a frozen tissue section onto the chip on the side. So we have a 1 centimeter by 1 centimeter standard chips offered in the kit for now, and also perform imaging with the standard microscope we have in the lab. And then the mRNA capture and cDNA synthesis will be done on the chip. And then we will be doing a rolling circle amplification to construct the cDNA library, and then later to be sequenced on MGI sequencers. So we also offer a end-to-end solution for the bioinformatics analysis as well. So for the comprehensive data analysis software suite, we are provided free of charge with the kit solution to be able allow you to go from image processing to reads quantification and eventually overlaying your gene expression profile onto the tissue section. So for our softwares, we are providing manual tools as well as automated solutions for cell segmentation. So for our automatic single cell resolution, we are providing an AI-induced machine learning algorithm that can help you achieve a single cell resolution with your tissue imaging sections. So our interactive visualization software is provided online and offline for you to be able to visualize the data and to do ROI lasso for further analysis. So currently, Stereo-seq was focused on biased whole transcriptome, but we realized that the multi-omics and multimodal analysis is something really important to making breakthroughs in scientific discoveries. We think the importance of traversing the central dogma of molecular biology helps you to answer questions about what a tissue was before, what are they happening right now, and what could be the future of it. So we are also offering Stereo-seq multi-immunofluorescence solution currently. So we are allowing a protein co-detection with a conventional multi-immunofluorescent assay. So this allows for high fidelity protein staining of the image and for enhanced spatial protein identification along with the unbiased whole transcriptome information. So we're able to demonstrate that the method is consistent with the already existing solutions we're offering, and we're also being able to detect up to 4 to 5 proteins depending on the channels of your microscope as well. But we are compatible with fresh frozen and PFA fixed frozen samples for this solution. We've demonstrated the Stereo-seq MIF solution on multiple samples. Here showing a mouse heart staining with some important biomarkers and overlaying it with the spatial gene expression profile. And there's more to that. We wanted to design a solution that can bring answers to address your spatial biology questions. And we are preparing to have these kind of features provided to researchers at the end of this year and also early 2024. So in order to facilitate the pathology research applications, we are combining H&E staining of the tissue section onto the Stereo-seq chip on the same slide. So for easier characterization of the spatial temporal heterogeneity for fundamental cancer research. And also we are combining with Cy-seq, which is protein multiplexing, combining with Stereo-seq whole transcriptome at the same time. So we were demonstrated that we can simultaneously profile hundreds of proteins at the same time and with unbiased whole transcriptome to be able to identify specific protein markers and also transcriptome markers at the same time at a very high resolution. And additionally, we are trying to answer questions within the immune microenvironment. So spatial immunoprofiling with TCR-seq and BCR-seq is something that works really well with Stereo-seq. So for immune cells that usually have a low capture efficiency on within the omic sequencing technology, we are trying to address this question by having TCR, BCR enrichment with our standard Stereo-seq workflow. So what's the next stage of Stereo-seq? We are currently in the pipeline, the R&D pipeline for the Stereo-seq FFPE, which will be released in 2024. So what does Stereo-seq FFPE offer to everyone else? We are still gearing toward trying to answer questions in the discovery aspect. So we're providing whole transcriptome profiling with high resolution, and also the probe design we have changed a little bit, designed for efficient capturing of degraded mRNA. And we demonstrated this technology to be very robust on low quality samples as well, such as DV 200 score at lower than 30%. Additionally, in order to embrace a higher throughput automation solution, we are bringing a STOMIX automation system into researchers' labs for you to process samples in a quicker and faster way. So this one, we are offering up to 24. And this is from post-imaging to cDNA collection, which works really well with our sister companies, MGI library prep automation system, and also the MGI sequencers. So for this machine, it's designed to improve the flexibility and scalability of Stereo-seq technology, and also to bring better automation solutions into their lab. So this provides an automated one-stop solution with intelligent robotic operating platform and software systems embedded within the machine. And we're combining the manual operation steps in the STOMIX automation system, so everything can be bundled together and you can do everything in a quick way, and then you can walk away and do some other experiments and then come back to check the results. And for Stereo-seq, we're continuously improving to bring better accuracy and accessibility to your lab. So currently, we're focusing on these 4 aspects as an multi-omics and a multimodal. Stereo-seq FFPE solution is definitely one of the most asked about, and we are trying to address this in a different way than the other technologies, hoping to bring more insights into your research. And for capture efficiency, for omic sequencing solutions, capture efficiency is always limited by the chip design. So we are upgrading our chips and hoping to increase our genes per cell, and also to reduce the cellular diffusion for better cell segmentation. And for flexibility and compatibility, we are providing large-sized chips up to 2 centimeter by 3 centimeter in the kit solution to be able to process for usually large organs, and also for smaller sized chips for tissue biopsy and organoids. And for our self-running algorithm, currently our cell segmentation algorithm is based on nuclear stain images. And for our upgraded Stereo-seq, which is also a paper we published in bioRxiv, with Stereo-seq, we're upgrading the cell segmentation algorithm and also including better molecule labeling and assigning transcript into the cells that they're supposed to be. So how to access Stereo-seq technology in Korea? We are happy to work with our distributor BMS, who also distributed our sister companies MGI sequencers. And there are several different options for you. We have full service mode and also kit mode provided around the world. So for service providers in Korea, and soon that we'll be able to find one and to be able to offer a full service solution for you. And as for kit models, this is also one of the solutions a lot of our collaborators consider because they have the full control of the entire process. And we also provide on-site technical support and training for free, and to
잘
make sure that
먹었습니다.
you can
네.
to make sure that you can
아.
work and process a workflow at your own lab. Yeah. And for if you're interested, you can scan the QR code and we will have a representative contact you very soon. So thank you for listening.